So the webinar has started.
Hello, everyone. We'll wait in 3 minutes so everybody can join the section. And after we start. So let's start. Welcome everybody to the EMN webinar series, today with the section 25. I'm Carolina de Almeida from Brazil. I will be the host today. And with me, it's Juan José Valdez from Mexico, that will be the co-host. We are EMN committee members. But before start the presentation with our speaker today, we want to share some advertisements.
So before we begin, we would like to promote our social media platforms. You can connect with us on LinkedIn, X, or Blue Sky, and where we regularly share events, resources, and important announcements like the call to join the EMN. Or if you have any questions, you can send us an email.
We also want to remember that we will have the Metabolomics conference on June 21, between June 21 and 24, hosted in Buenos Aires. So it's the first time that a country in Latin America will host this kind of conference. So if you want to join us, you can register yourself in the Metabolomics webinar link, sorry. So metabolomics2026.org. And you can also submit your work if you want as poster presentation until May 14. So please join us in this conference.
Also, we would like to take a moment to highlight the upcoming EMN virtual networking event which is titled Developing the Scientific Identity as an ESER in Metabolomics and Beyond. This session will feature excellent panel speakers who will share their experience and perspectives on building a scientific career. The event will take place on a few days, so you have still time to register, and we warmly encourage you to register, and we will see you there.
So now we can start, and before Juan share Diego's biography, I want to thank you, Dr. Diego, for accept this invitation. Today we will have this really interesting talk about a different team, Archaeometabolomics, the new way to delve into the past.
So today I have the pleasure of introducing a Colombian researcher specialized in the field of archaeometry. He holds a Bachelor in Science in Chemistry from the National University of Colombia and a Master of Science in Chemistry from the University of Los Andes, also in Colombia. In addition, he completed an Erasmus Mundus Master in Archaeological Material Science, with joint training at the University of Évora, the University of Thessaloniki, and Sapienza University of Rome. He earned his PhD in chemistry at the University of Florence in Italy, with a focus on science applied to cultural heritage conservation. Throughout his career, he has built a strong interdisciplinary and international experience collaborating with archaeologists, curators, conservators, and other scientists in both laboratory and museum science environments. As an expert in archaeometry, he applies scientific approaches to address historical, artistic, and archaeological questions, contributing to a deeper understanding of past societies and cultures. His current research focuses on the study of cultural heritage of the objects using modern analytical techniques, covering both organic and inorganic compounds to a microscale or in situ analysis, by applying rigorous scientific strategies to extract and interpret physical-chemical data. He's able to generate precise and meaningful information about the materials under study. Currently, in the Tobacco Health and History Project, he is responsible for developing and implementing metabolomic strategies, including analytical protocols and experimental approaches to investigate the molecular chemical information related to tobacco consumption in past societies. So without further ado, I leave you with Dr. Diego Armando Badillo-Sanchez. Please.
Hi, everyone. Thank you so much, Carolina and Juan, for this invitation. I appreciate it a lot. It's an honor to be here with you all talking about this almost new field that's called archaeometabolomics. So I'm expecting that you have this new interesting topic in your field in the next years, and you have in your research this kind of mind that is possible to go ahead in other directions than the traditional ways of using metabolomics in clinical and life sciences. So I'm going to start to share the presentations, okay? Okay. May you see the slides?
Yeah.
Yes.
Perfect. Perfect. That's great. So in the next hour or so, I'm going to speak about this new technique called archaeometabolomics. It's not a new technique for you. It's a new representation of how we put together two perspectives of a study. So in this presentation, I must to disclose that can be some slides that can be disruptive for someone because it has some images of human remains. And I took the freedom to create different figures using AI to increase the platform as this is recorded and later is going to be reviewed for other participants of the Metabolomic Society. I hope that they take care and put attention to these slides and more than my single words. So today I'm going to talk about archaeometabolomics as a new way to dive into the past. This lecture is going to be a step-by-step structure talking about the archaeological sciences. What is the concept of this archaeometabolomics idea? What are these analytical limitations? What we need to do and what are the methodological requirements? And later I'm going to show you some cases, some applications to see how it is used, to end with some future directions to what we need to go ahead to apply this archaeometabolomics in a deeper way. So to start, I must to say, okay, Juan, present my bio very well. I am a chemist working mainly in the cultural heritage field. So in 2019, I was doing my doctoral exchange at the University of Los Andes. I was working mainly with silk historical textiles. So using different analytical platforms to study these materials, I was focusing using high resolution liquid mass spectrometry with liquid chromatography to understand different aspects, and some of those were the dyes present and how they were evolving through the time, how this has a molecular repercussion to degrade the material and affect the history of these objects. I start to notice some patterns in there that traditionally we only focus in archaeometry in single dyes, one molecule, but when you open the broad directions of what the new analytical tools are giving you, you start to see some patterns, some ideas, and by chance, the same year, the metabolomics core at the University of Los Andes, they had the first core metabolomics school, and I was able to participate next to Dr. Calab. It was a fantastic event. They opened the center, and I start to see another direction of how metabolomics can be used. What is the meaning of this field, and what is the purpose to explore large numerous of molecules at the same time? And that put it together with historical materials, what can be implicit in there? How can we start to dig in to the past to find new words that we cannot describe until today? Then 2020 hits, pandemic situation, and at the end of that year, I started to work with the Tobacco Health and History Project at the UK University of Leicester in a fantastic group led by Dr. Sarah Iske. She had the idea to use metabolomics to find, if it was possible, to study, if it was possible, the tobacco consumption. So I was there for the next 3 years until 2023. So the result I'm going to present you is about that period of time, where we investigate metabolomics in this archaeological world and how we put it to work together to find what does the meaning of that. So usually I have been working in metabolomics applying to archaeological materials, but from the perspective of humanities. So at the beginning of my talks, I need to start saying, what is metabolomics? What are the techniques? But today, I'm going in the opposite direction. I need to introduce you what are the archaeological sciences, because we are, from the scientific perspective mainly, here in the Metabolomics Society, we are more familiarized with life sciences, with pure science, let's say. But what are the archaeological sciences? What has a meaning? So the archaeological sciences is a combination of knowledge. It's the new interdisciplinarity that is created to put in context different materialities. It's a way to create a perspective that allows to archaeology, history, humanities to understand what was happening, how was happening, and to create new ideas. So sometimes archaeological sciences, as well known as archaeometry, are kind of the same thing. And we use different techniques using analytical platforms, spectroscopical, microscopical, MS, isotopes, different kind of things to analyze different materials, both organic and inorganic ones. So it's not centered only in one question, in one perspective, but it's a broad way to research, putting together what was happening in the past, how we can study that information. So in that way, we help the traditional archaeology. Archaeology is looking at time, the evidence from all different periods in the past. So we, as scientists, give a hand to understand those objects, those materials, put a new perspectives, clarify ideas, and integrating that with humanistic questions, we find the core of the meaning of what was happening. We can integrate that to create ideas, impulse new theories, and of course, we all have the knowledge, have read about, for example, evolution. We know that it's a trending topic sometimes, but in the real matter is a lot of work between different disciplines, looking and understanding and changing the ideas constantly. And that has an impact, not only academically, but in the real perspective of humanity. So the scope of archaeology is to reconstruct the past. And together with archaeological sciences, we start to look at with more detail. Sometimes we have some perspectives in the humanities that can look ambiguous or can look only as a theory that has no explanation. So the sciences start to dig, to put to talk those subjects and to trace the information that can be used. Of course, we have the historical record, but the historical record, we have some issues with that because it's not for all the period of time. Has changed. We have some literature problems. We have some visualization of what was written down, how we interpret that, who wrote it, and not all the information is in the documents. Even some documents need to be studied harder to interpret that. So in combination, we integrate that to put a knowledge that is not only to talk about the past, but allow us to understand the present and visualize the future, how it has a trend, and how we can create a real humanity understanding without limitations. But of course, this is very difficult when we talk about time, because it's not stable in time. The material has some degradation, change. All time are changing, and not only in the material constitution, but as well from the culture perspective of the meaning. So we need to start to think about different aspects, not only about the materiality, the object, the composition, of course, is very important, but about the meaning and how that creates ideas, representations, and how are used or how were used, because that creates a different impact. Simple example. If we talk with new generations today and we talk about telephone calls, we always are going to talk about cellular connections in the library, not those old big boxes that in the past we have connected by line. And that has some ideas that are changing. So the materials suffer changes. One very important is the degradation because the interaction with the surroundings, by the interaction with the atmosphere, with temperature, with humidity, that change the composition, degrade it, sometimes make them disappear. And we have as well the interaction with biological agents. So we have another interactions that are coming there, putting some molecules, taking out some molecules, and what we have at the end. How as scientists that we are involved in the molecular world, what does meaning that? So let's put an easy example, the most relevant for us as humans, our bodies. We are alive, yes. We have a name, we have an attitude, we have a plan, we have family, we have surroundings. We live in different cultures and interactions. But at the moment that we die, we decompose. We go in a series of changes that creates a disappearance of our materiality. In long terms of time, what we have at the end is not what we were at the beginning. And of course, we cannot talk with them. At the end of the time, if we have some bones, let me try to find here the pointer, sorry. So we start with a body, yes, full of tissues, full of processes that start to degrade, goes in decomposition, and bacteria, fungi, and other agents start to interact and are replaced and move. But what is that information that we have at the end? What we can interpret from those bodies? Only the macroscopical information that we've obtained there? Or maybe we can start to track some molecules that allow us to get more information from the past. For that, we have two perspectives. One is the archaeological, and the other one is the forensic. Both are similar in perspective, but in time scale are completely different. Forensic analysis are focused in the short time scale, very short, days, months, some years. Meanwhile, archaeological perspective goes beyond some decades, centuries, or even millennia. And that has a repercussion on how those materials are degrading, are changing, and how we can extract information to track the knowledge from those objects. The objects are not only, of course, the bone itself, can be any kind of material, can be a stone, can be ceramic, can be a textile. We have a long list of objects from where we can investigate and obtain information from there. The traditional approach from archaeology has been exploring the macroscopic figures, what we can touch with my hands, what we can see with our eyes. So we go, we dig a field, we found some archaeological buildings, we found some mortuary practices in there, some objects, and we rate them. We have something that is called stratigraphies, that are the different layers of the position, and we have an archaeological context, that is how each object has a relation between among them. So, for example, if I found a coin, how that coin can be related with a body that is in the same layer, and I can do some traces in time because that coin must be manufactured in a period of time. So in the previous time doesn't exist. So give me a correlation in there. In the same way, looking at the bodies, there is some features that we can observe. We can see diseases. For example, if there is trauma, there is an infection, there is a change, there is a break. We can use some techniques to find if the objects of the people were moving different places. But that's in the macro scale. And that gives us a perspective, a view. But do biomolecules persist after deposition and subsequent degradation? Maybe we have some molecules that we can investigate to give us more information, or only we have these macromolecules, these shapes. We have a shield, we have a base, we have a pot, and only by shape, by color, we can give ideas. So the analytical chemistry goes and starts to check. What we found? Voila, macromolecules survive. Big molecules are big enough to survive on time, even by millennia. Some approaches of that is, for example, the zooarchaeology by mass spectrometry, more known as ZooMs, where investigating collagen and other proteins, we can understand what kind of a species is the bone or was involved in a preparation, or by paleoproteomics, understanding different dietary composition. Of course, ancient DNA is a beautiful approach that we are understanding. For example, that it was not that traditional linear evolution that creates in the old school idea. Oh yes, the humans come from the monkey and goes in a line of evolution. No, it was a branched one, and living together different species of hominids in the same area. But what about the small molecules? How is that transformation? Those survive in time? Or almost be very big to have a time scale to be studied? We know that during time, there are different processes that degrade the organic matter. Big molecules suffer some degradations, and we can find those traces, and by new computational methods, we can put it piece by piece together and understand it. But the small molecules are all consumed by this degradation, are all disappear, so we cannot talk about anything about that. Here is where Archaeometabolomics go ahead and say, no, must exist an option to study these small molecules. So Archaeometabolomics goes as an analytical solution, that is coming from the life sciences to the archaeological world. And what is the implication of that? That we need to have a sensitivity by the nature and the context of the samples. So we cannot only go ahead and perform experiments, we need to think and create a new way to think what I'm doing. What is that? So we bring the metabolomics applications to study the archaeological materials. In the analytical perspective, this is very similar to the traditional metabolomics that you do all the time. We have almost the exact framework. But we have a specific 2 points that are completely different. The start and the end. The experimental design must be more rigorous. We need to be more exigent in what we are proposing, creating a workflow with more robust analytical strategies to have at the end an interpretation. Must have an archaeological context interpretation. That information that I got at the end, I can use it because it has an impact on how history and how the context can be seen. So we use almost the same platforms as GC. To be honest, have not been very deeply used another analytical techniques, but we put a stricter contamination control We are cautious with the extractions, and we look by the taxonomy, and we are aware about the interpretation. So we need to think twice the experimental design. Why? Because the abundance of samples is not as in metabolomics. We have uniqueness. We are talking about samples that the metabolite concentration is very, very reduced, ultra low concentrations. So the homogeneity of the samples is not simple. We have covariance in nates that is very difficult to understand from where it's coming, that can give us some ethics difficulties, because, okay, you are talking about very high valuable objects. So why are you going to touch it? Are precious, those samples. Those samples are not a traditional experiment that we use. And we have a large variability that sometimes we cannot eliminate everything, so we need to be aware of that. But what it means uniqueness? Uniqueness is because sometimes we literally have one single object, one single individual that cannot be replaced, cannot be sampled twice. We can have uniqueness because the organic preservation is only that object, because their context, their environment, their archaeological context is only one place. Only one cave in one area of the earth contains those individuals that we are going to sample. Only one material has a technological trace expressed in the past, cannot be replicated. So we need to take the time way ahead before to decide what we are going to do, to think about that, to take the permits, to know what are the impacts, what are the repercussions, what are the limitations of those samples. And that creates some analytical limitations, of course. Preservation. We have diagenesis, transformations, mixed things, because the objects that come from the past they have a long biography. And we need to take care about that. How was created? How was used? How was transformed? How was discarded? How was found again? How was collected? How was stored? How we are going to sample? Too many questions, I know. And that gives us a material access, because are too sensitive. Sometimes we are not allowed to take large samples, so we cannot have replicates. We cannot do whatever we want with the sample. We can have contamination from different sources. And as we know, in metabolomics, that's an issue, because we are using ultra-sensitive instruments, and we can see almost everything. And that's the good thing about archaeometabolomics. We are approaching that. But for that, we need to take higher quality assurance and quality control processes. There are no standard protocols. This is new materials, never investigated in the past. So we need to take more time to establish what is going to be, to validate those methods, to know how it's going to be that interpretation. In that way, we are not going to have overclaims. Because again, it's important to highlight that the goal of an archaeometabolomics process is the result, that interpretation. How a society was in the past, what we're doing, what that has a meaning of the past. It's not only about what is a biomarker. Okay, I have 2, 3 molecules, but what is the implication? With that in mind, let's talk about how it's used in archaeometabolomics. So we are going to focus mainly at the beginning in humans. So it's the most appealing approach and most interesting from my perspective. So we know that we die, of course. We have ceremonies today that can go to the grave, but that has a decomposition, has a change of the nature of the original metabolites, and that's changing in time. And when we talk about archaeology, it's not exactly the same. Where have been gone all those molecules? Have been already disappeared? Transformed? Are there? There were thousands. Where were the metabolites in the flesh, the muscles, tendons, brain, lungs, everything? We usually only find bones. In the clinical studies, we are very lucky. Come on, you are next. Give me your sample. Okay, we have a mistake. Sorry, we need to repeat. But you come in 1 week. That's great. In the Archaeometabolomics now, we cannot repeat. We cannot go ahead to change what was happening. We cannot discriminate about the questions that we do with patients today. So we claim that all body is a continuous fluid of changes of the metabolism, and that goes to different parts of the body and in the bones. That is what we found at the end as archaeologists. Has even some capillaries that goes to all the directions of them. Even in the marrow, we have the flow of those metabolites. But they're not the same, depending on the age, the parts of the body are in constant change. So how we can compare that? How we can trace a statistical study that is going to allow us to create a pattern in there? So after that, we start with a complex mixture of tissues. Okay, we have some bones, the marrow inside, tendon, flesh, and everything. The gradation starts to occur. We have a kind of a soup body in there, a lot of microbiome. And we found a solid, empty, hard material. Where have been gone all these molecules? We are familiarized with the permineralization process when we talk about fossils. Oh, yes. We have the exchange of minerals, and these materials are changing in the structure and the other hydroxyapatites, and we have that solidification at the end. And the organic molecules, we have the same process, and we have a flow. We have a direction depending on how the surrounding is creating that degradation. So we look at the bone, this hard material, and we look inside, and we start to detect different particles, of course, that are coming from the soil, salts, everything. And usually we think that is only that, right? A hard object. When we look in detail, analyze what we are having there, for example, these little clumps are the reduction of that marrow, red cells and everything, and are clumps of iron and magnesium closer together. It's okay. That gives us a clue. Those cells were present. They are drying up. They are ending in some parts. And we look and we find several variables. All these 4 pictures that you can see here come from the same bone, one single bone. And you can see different things around that. What are those? When you start to look in detail, some are coming from the ground, yes, some soil there, but others are the remains of that soft tissue that even disappear completely in other parts, no. And that's complicated because what we're having there, a single material, a mixed material. So we know that bones are usually considered as a compact material. Almost 70% are mineral. And from there, in the decomposition of that, we have in fresh bone less than 0.05 to 0.3% of metabolites in fresh material. In archaeological bone, reduce a lot. But we have two parts in bone. We have the compact bone and the trabecular one. Is the same? Should be. What's happening there? The thickness, depending on the bone, is going to change. And as well, the relation between trabecular and compact bone. So we have a native covariance in there, depending on what type of bone we have and what bone structure we have. Is the same a rib, a clavicular, a vertebra, one bone from the hand, a femur, and one single bone, you have different areas as well. So we developed a technique to study that, to check what was happening in the different areas of the material. I'm sorry to talk about humans in the past as material, but please take care about we are only using it as a purpose of investigation, and we take all the consideration of these subjects, of course. So we have there the trabecular material, and in the bond with the what we call it the compact bond is really composed by different layers: periosteal, endosteal, cortical bond. And as you can see here, when you sample each one, you start to notice that you have different textures, different colors, different material. And as chemists, it's a very simple principle that if you have something different, the composition is different, the chemical information is different. So we tested there, and we found with one principle that to study in an archaeometabolomics perspective, human archaeological remains, or HSR, those are made of organic components, so must have metabolites in there. But from where? From where is going to be preserved those metabolites? So we took two perspectives. One are the remains in the trabecular bone that are coming from those old remains from the marrow. And in the cortical bone, all those cells coming from the bone remodeling process. As you can know, the remodeling process is a continuous process, all the time. Meanwhile, the living organism is alive. That process is going, can be interrupted or disturbed depending on diseases or other factors. And we have some osteocytes inside trapped in there. Those osteocytes have information that are very well protected by that cage of hard inorganic tissue. So with the help of modern analytical platforms, will allow us to detect those ultra low molecules. So we have the bone, we have the cortical bone, taking out the periosteal part that can have soft tissue, soil, and other information. And we took the sample from that region that is protected, almost protected, and from the trabecular sample. We create that kind of sampling to create micro drilling, and we select femora to study and homogenize the study. And at the end, we have very similar samples. In texture, sometimes, depending on the degradation of collagen, but in coloring and other things are very similar. With that in mind, we develop and establish an extraction protocol. For what? To have polar and less polar molecules from a single sample. Why from a single sample? Because we have unique samples. We cannot go and take one from these individuals. So we maximize the amount of information that we can extract from there. Usually in metabolomics, what we do, we create a list of patients, individuals, samples, create the questions. Let's check who is going to be present, who is not going to be. We don't. We go to different process. This picture, I love it. It's one of the campaigns from the collection of the individuals for all the treatment and analysis. And this is a chapel in the UK. And as you can see, the individuals are displayed one by one, and it looks nice as with the respect that they deserve. They are going to be treated as a patient. Even 500, 400 years later, we cannot go and ask them the same. But we are going to be completely aware of what they can say without words. We perform all the sampling, transform that in extracts with a long list of QCs. We must to create QCs for everything. Pool QCs, individual QCs, from the ground, from everything, to take out all those biomolecules that are not relevant for our study. So let's see what happened when you do that study to check about tobacco consumption. That was our goal in the THH project. So we studied 323 individuals from 2 locations in the UK. 1 cemetery in London and 1 rural area in Barton. 2 populations, both with post-tobacco introduction. Why post-tobacco introduction? Because tobacco is not naturally grown in Europe. That was one of the consequences of the interaction between Europe and the Americas. When the Europeans arrived to the Americas, they find the natives using some plants, and they start to look for that, and they move it and create a big commerce that is still in course. As you can see the individuals, but the color of the bones, they look very different, very, very different. Why? Because the ground, the soil, the environmental conditions of degradation are not the same. So is that going to have an impact? We are not going to have anything, statistical representation there, and only going to have very dispersed data. From the rural area, we took some pre-tobacco individuals to have, a section of the group that is going to indicate us if you are finding tobacco, something that never used tobacco, you are doing wrong things. Come on, check it again. But those individuals, will start to listen them through the traditional socioarchaeological analysis. In that way, we're classified in 2 groups. One group known as tobacco undetermined. There is no dentation, so it's not possible to study and know if they were using or consuming tobacco. Other group with full dentition, no modification, perfect, group as tobacco undetected. And the third one, they have some lingual staining on the teeth and some pipe notches, indicating that or they were used consuming tobacco by chewing or smoking that created this lingual staining, or by smoking by pipes, creating the abrasive erosion on the teeth, creating these notches. They were classified, and we have almost 63% of the rural area individuals as undetermined, 40% in London as undetermined. Classified by sex, or biological sex, I mean. And we have these individuals in range of time between 1500 to 1850. It's a very large span of time. We apply the analytical methodologic workflow. What is our archaeological contextualization? To understand if we can detect tobacco to see what was happening in that population, of course. We do the extraction that I explained before. It's a repetitive sequence of different solvents to maximize the extraction of those micro concentrated metabolites. We have that process for helix separation and C18 separation. And we applied it in our LC-HRMS analysis with some batch, of course. Each one with a nice sequence of control. In that way, we can check, of course, the analytical platform, extractions, everything, and be useful later for the data treatment and interpretation. Because if not, it's going to be very chaotic. What we have at the end, voila. We could separate the groups between detected and undetected individuals. That means consumers of tobacco or not consumers. They'll give us some information. 125 less polar molecules with 44 molecules that are highly relevant. But at the beginning, when we start this process, we have more than 1,000 of features on our datasheet. From those to arrive to these ones, we do a lot of cleaning, rigorous cleaning, to be sure that only the biological relevance information is going to have from here. And not, for example, the pesticides that were found. Of course, the two groups from London to Barton rural area are different because they have different agrochemicals, different ecological systems. But we took the time to do that. With those models of detected and undetected, we applied it to the undetermined individuals. We create an algorithm to check it, and to test it, we use the pre-tobacco era people. What we have is, for rubber phase measurement, almost 90% of precision. By the helix, 75. So here's where you stop and think, okay, but what is happening in there? What is happening with some molecules and the other no? And it has interpretations, of course. As for example, that some molecules move different through the soil with the water. So the polar molecules, respectively, apolar ones, have different proportions of exchange. And that moment that is in equilibrium is going to have a repercussion later when we measure that. So what propose is, okay, you have two ways to look, test it, and if both say that they have the molecules that are classified as consumers, you can say that, okay, was consuming or not. So in that way, we combine osteopathology to check the physical or documented evidence of diseases with our archaeometabolomics information. We look for those metabolomic markers from the organic matter bonds to establish the damages produced by the tobacco consumption in humans. So Dr. Anna Davis Barrett, to the archaeologist that did the study, she checked by these individuals, applied the model, and we have the large set of different people classified on not only by biological sex, but as well by age and by economical status, to check it against diseases. For example, maxillary sinusitis and pulmonary inflammation, to check who were doing this tobacco consumption and what were the repercussions of that. When they looked at that, they found that despite King James I's cries against a costume loathsome, tobacco use became an intimate part of post-medieval English life, consumed by all facets of society. And that's very important, because that gives us a view of how was that society. Female and male were using tobacco. Even young people were using tobacco. And that has some repercussions on the health. The cumulative impacts over time of poor air quality tobacco consumption and other risk factors appear to have affected Londoners with an increased likelihood of the presence of maxillary sinusitis in later age groups in this population. Yes, we can start to look how was the society the health, and that can give us information about the economy and how the society was moving. So we use archaeometabolomics to create a narrative about the past. This was applied as well to other materials. We study tobacco from this nice set of historical tobacco and compare against modern tobacco to evidence some changes in the different plants. What we found is that we can use those archaeological materials that are stored in collections for centuries and still perform a metabolomic assay to understand them. As you can see here, NTNR Nicotiana tobacco, Nicotiana rustica are two species of tobacco plants, and we have here one sample that it seems very confusing. When we took out from that, the two cluster groups are discriminated beautifully. Why are they split among the different samples? Because as well, the ground location gives differences in metabolites, and we can find here from Cuba to another Caribbean regions and discriminate about that. So, okay, we had in the previous slide one sample that was not fitting the theory, right? If this sample, number 8, is classified as Nicotiana tobacco, we applied our models, and we found that with this almost 80% of accuracy, it's Nicotiana rustica, not Nicotiana tobacco, as it was classified in the catalog. Meanwhile, another sample, this one in F, that has not any classification, is almost 50-50, almost 60% Nicotiana tobacco, Nicotiana rustica. Why? So the model is not perfect? Yes, kind of. So when we compare it, the results with the reality, yes, this Nicotiana rustica looks very similar to Nicotiana rustica. And this Nicotiana tobacco looks very similar to Nicotiana tobacco. But it's probable that this sample that was not classified as rustica or tobacco is because it's a new species created by human. Because it's from the time that the people start to breed them to have the better plants, to have longer leaves, to have better productions, to be more optimal for the crops. So maybe for that. And that gives us new perspectives for the historians, open new ways to discuss the past. Another application was a study about Yersinia pestis. Yersinia pestis is a black pest in Europe that kill a lot of people. So we use daytime to study it was possible to classify between people that was infected and not infected. And we compare this data with genomics one. Unfortunately, as you can see here, there is not representation between both groups. Now, would you say, so, okay, so you cannot use always archaeometabolomics in there. When we look the data a little closer, we found a little subsets of individuals, and those were more related with the age of the individuals. As you know, depending on the age, the development of it is different, and the remodeling process as well. So that can have some repercussions in there. And we were using, let's say, a second-hand sample from the genomics. So we're not an experimental design direct for archaeometabolomics, was a second-hand. And we have in there that repercussion in there. For that, we create new protocols to sample dentine, trying to check that what area is the most relevant to have that organic information. Because from this set of data, we took the root of the teeth. And the root is mainly inorganic, cementium and other things. Meanwhile, the clusters of organic material are preserved in the center. But of course, this is very destructive. And not all the people is going to accept that unique samples are going to pass through this. So our studies in archaeometabolomics have some impact lately, have been more noted by the community. Our study to characterize phenotypic differences in human cortical bone was a nice discuss and public advertised in different medias, was almost 1 month in all kind of languages around the globe. But of course, this is not the end. But archaeometabolomics has a future. We need to create this technique and put it as a routine technique, decreasing the cost, decreasing the barrier between the scientist and the humanities. We need to have both the same sensitivity, the understanding. We need to increase our libraries. We need to update all the artificial intelligence assisted annotation, because we have a lot of biomarkers that we need to check in there. We need to create the data sets. We need to establish the protocols and make from this approach a routine, as it is today's proteomics, genomics, or ancient DNA. In that way, we are to consolidate all those to create a new language about the past, decreasing the speculation from the narratives. In the future, we hope that the nice technologies will allow us to work, to find the biomarkers, to understand what are those, from where are coming, and no matter what kind of material we are using. I'll repeat again, I showed you here mainly from the human bones because it's the most interesting, but other groups are working in potteries, in organic residues, in other things. So some takeaways ideas to you. Remember that the small molecules in living organisms provide insights into their lifespan, and we can look on that. But archaeologically unique materials require training, perspective, sensitivity. We cannot apply our life sciences experiments straight to the archaeological material. It seems similar. Both are the same analytical instruments, but we need to take the time. LC is a good approach with the new computational tools. It's allowing us to reveal the past. And the use of this high resolution allow us to understand the past and create this language. But we need to create the force that you in your labs open the opportunity to archaeologists to say, yes, we are going to do it without the fear that, oh, you are going to contaminate my instrument. That one answer that I received some years ago. And today we are proving that there is no damaging in the instrument. In the opposite, we are creating new data. But we have, of course, these technical, operational, and administrative challenges that we need to resolve. Of course, are not cheap. Experiments has a cost, but the final cost can be paid by the understanding of our past. I must acknowledge to the Metabolomics Society and the Early Career Members Network to this invitation, and of course to my previous group, Tobacco Helen History, led by Dr. Sarah Inskip, with all the team, and to Dr. Monica Cala, who appreciated inviting me to this talk. If you have questions, I'm very open to follow up. Thank you so much.
Thank you, Dr. Diego. It was an incredible talk, a completely new team, but so interesting. We are open now for questions, so please, for the audience, if you have any question, feel free to send in the Q&A box. But we have some questions, but the first one, it's my question. I have an question about the ethical considerations. For example, when you find some archaeological material, how much you can explore this material? Because when I think about this kind of material, I think about the preserve cultural material for the museums, for example. How much you can explore, do the extraction and analyze this kind of material without cause more damage for bones and other kinds of human materials.
Thank you, Carolina. The study of the cultural heritage objects is very tricky because you need to start thinking about the object or remains in a long perspective, and with a goal that those objects must to be survive for new generations. So it's the most valuable part. So even if I have a good idea and I have a nice instruments, but I'm going to destroy everything, it's not going to be possible to do any kind of experiment. Yeah? Because you need to survive that object for the future. Maybe you were wrong, you don't know. Or maybe in the future there are more valuable instruments, more sensitive ones, and can give you, and you need to wait. So as I was talking about the biography of the objects, we need to talk about them with that knowledge, and that creates a limitation. Because with archaeometabolomics, you need to understand what kind of molecules are coming out from your sample and what that means. Because you can have a nice molecule that gives you, okay, this is the most brilliant, that is going to explain whatever process that you want, but you need to be sure that was not introduced later for some kind of contamination, for some foreign process, because there was a co-layer deposition of another individual that affect yours. So it's a interdisciplinary team that must take that part of the experimental design and take the time, because sometimes I have I had the opportunity to review some articles on people, and as this is new, they have not understood the sensitivity that you need to gain. Because that way, the sensitivity that you need to have is what are the limitations? What are the limitations of your results? What is the limits of your interpretation. And not say, because it's another barrier that we have between humanities and sciences, that sometimes humanities takes the scientific result to put as a true. And you cannot deny it because I did a scientific analysis, so it's the truth. And no one can say other thing. And it's not that way. So we need to balance. We need to discuss that. So yeah, it's tricky with the cultural heritage, and more in archaeometabolomics because you need to create the composition of the groups. Because if not, you are jumping to nice LC experiments, for example, or GC experiments. Yes, if you have only one sample and you have thousands of molecules, that's not archaeometabolomics, truly. Yeah. So if the understanding of what is metabolomics, what is archaeology, and put it together, but with the sensitivity of the sample. Because as you said, whatever can happen with the object and must be preserved. And the other thing is that, for example, we think today about museums, but museums are only custodians of the objects, are not the owners. The owners are the humanity. Yeah? So it's not from one country or one museum, it's from all. And we need to understand that only collectively we can create that understanding.
Okay, thank you for your answer, Juan. Don't know if you have some questions. We have questions on the box. So you
Oh,
think we can...
yeah, we can start with the questions on the box. I'll ask mine later, so I have a lot of questions. So, so I'm gonna start with the first question that is in the box. They're giving you congratulations first, though. That was a funny say. The question says that was a fascinating talk. Thank you for it. And the question is, can we use archaeometabolomics to discover other types of lifestyle in our ancestors? For example, diet and exercise? If so, how?
Yes. The answer is yes. Completely yes. We can start to look about the past, but we need to create the groups very well defined to check what was happening. And when we combine that, the archaeometabolomics, with the forensics part, we can create, for example, the group from the forensics people that we have in our institutions in different parts of the world, that we know their daily life, and they are there collected by different purposes. Yeah? And we can use that as a training set and later test it in those archae samples. So there are different approaches, but of course we have here the legal and ethics limitation. So the most important there is that part. So we need to understand that we cannot jump to take some population, for example, I'm going to test, no say, Colombian versus Brazilian individuals and check if they are more sportive, and only I'm going to test it without a rational. So yes, we need to cluster that and to understand what are the numbers that we need, what is the implication, the statistical impact and everything in the experimental design.
So the next question is, what was the biggest challenge you faced when applying this technique? And I will add another question that is similar. What analytical platform you use it? Do you need the newest platforms to do this, like the astral spectrometer?
Okay, so the biggest challenge was to intermediate the language barrier between humanities and science. So at the beginning, I had to discuss and convince the humanities that the bones were not only solid materials. Because for them, at the beginning, I say, okay, look these spots, and what I showed you in the pictures. You have there the soft tissue that is remaining. And they say, no, this is discoloration because the ground has some soils and has oxidation. And so created that, okay, you have there some organic molecules. They always think about for example, in the bone, with a hard material, and later to explain you have different layers with different information was chaotic. Because they say, oh, no, it's a bone. Take a chunk of that, place it in the machine. So no. And later with the sciences to explain that, okay, we validate it, we create workflows, we are sure that the data is true, and we take out all the other misinformation. So convince both sides that what you are having is real was the most challenging part. But it's a language barrier. It's because we are usually, for example, you do your RC, your CGC, you have thousands of counts, right? And you have your information in there, whatever you want. And we are jumping to only above the sensitivity of the instrument. So we are only in the threshold of what we can really check. So that's analytical challenging, yes. But it's mainly to discuss and to talk about, for example, the process of data treatment. Oh no, you follow the traditional data treatment with MC-MINE and you delete everything. Because of course, what you have at the end is only the signal from the soil that has the big peaks, but the biological information will disappear. So no, you need to change it. So it was a big challenge. So we're 3 years, and mainly thanks to the pandemics, so I cannot go outside. That helped me to that. And respect to the instruments, we tested, you can see there one of the publications, we have our water cell C mess against the Orbitrap. Of course, if you have better instruments, you have better results. Because what I was talking about the sensitivity. So you have there, we use IMS, ion mobilization systems to split the molecules, and that helps us a lot. So yeah, that helps. But depending on the limitations of your current equipment, you can propose some experiments, but you need to take care about that. So let's say that with the current technology that almost everyone has, yes, you can do it.
Awesome. So I'm gonna head with the next question. It says that I'm wondering whether there are any studies that analyze dinosaur fossils. So dinosaurs.
Yeah, with dinosaurs, it's very funny because we have a very long discussion in the group and with the associates, because we create the word archaeometabolomics, and the others want to put paleometabolomics, and that creates a breaking time of thousands of years. Yeah. So we have no tests yet. Fossils, some of them have tried to check some mammoth things and Neanderthal, but not so old to say that with millennia you are going to have information enough. Maybe we don't know, but as well, we don't have the number of individuals, nor the question to why are you going to sample the dinosaurs? What do you want to look there? And so, yeah, but this is that dinosaurs, no, not yet.
So the next question is, what is the biggest source of uncertainty that in the field that you think it isn't still underestimates? What do you think that it's underestimates in the field?
I think that the biggest we tested is the problem about the collection of the samples of the remains. because when you have the deposition of the remains in the ground or whatever archaeological context that you have, they arrive to a stable chemical composition. But that is disturbed when are collected. And sometimes we have two ways of perspective in the archaeology now. One is the old school, that I took something, I put away as I want, I don't care. And the most modern, that take care of everything, what was happening around, what is the context information, and what bag are you going to put it, because you have, of course, this plastic contamination. After, if you are storing in one place that were smoking or in contact with food, you have that an impact to the results. So that's the biggest uncertainty that we have, because some people don't check for that. And you say, okay, I only have an historical object from the museum and I'm going to test it. But okay, what are your QC protocols? How you are sure that the information is correct? So it's not underestimated. It's only about, again, about the sensitivity that we need to create in this specific field to be sure that we don't have more information from what really we have.
No, so I'm gonna go ahead with the next question. So it says, what kind of questions about the past do you think Archaeometabolomics is fundamentally unable to answer?
Unable to answer whatever that we cannot create good groups of study. So if we have an idea about whatever, but we cannot create those changes, statistical changes that can confront those ideas, we cannot. So mainly that.
And in the other question, in our experience, what has surprised you the most when comparing ancient metabolomics profile to modern ones?
There are different things. For example, from the tobacco, historical tobacco, that is a collection that is stored in London in the Kew Garden for more than 100 years in the same place. I mean, because we're collected 300 years away. That the molecules related with modern samples are very close. So allow us to create, for example, models about the past with present composition. So we can create, if you say, for example, you have a pot with a preparation inside that you don't know what were inside, what were these people eating. You could recreate it with modern recipes, test it, and after go back. So that creates another way to check it, because we have human changes in the nature. So we have changed the plants, we have changed the animals, and that have metabolomic changes, and we can check it. So for me, that's very surprising.
Yeah. I'm gonna head to the next question. It says, do you think there are cases that this field could mislead interpretations rather than clarify them?
Yes, of course, yes. Mainly when you don't have a good quality assurance process. So anytime that you create an idea, a research, that you don't take the care of protecting your data, yes, you are misleading. And you are going to have the impact that other people can say, okay, but check this point, check this out, what was happening, you don't check. For that reason, we need to think twice the experimental design. We need to be very sure that at the end, the result that we have is giving new ideas, are plausible, and of course, can be compared. For that reason, we did this analytical extraction process for two experiments, the Helix and the CXT. okay? Because if you compare two ways of measuring different molecules with the question, and at the end, you arrive to the same, maybe you have a bigger chance you did the correct things. Yeah? But if you only focus in one kind of molecule, and this is the tricky part when you do, for example, targeted analysis, that I forgot to mention that. We focus in on targeted, but you can do targeted analysis in archaeometabolomics as well. And that's as well another perspective that you need to be very sure that why are you targeting? Why?
And I think now we have the last questions. Thank you for the amazing presentation. Do you think this field can challenge current interpretations on archaeological sites?
Yes. And it's not because I'm working on that, but because we have the discussion with different people from the archaeological sector, from different scholars in different conferences. We talk about what is the impact of this. And this is challenging for them because as well they create some narratives with some speculation, of course. It's part of the humanities. And these interpretations change how you can create those narratives. If you have a molecular way to confirm your idea, you should try it. Yeah. But yes, this is challenging and creates the perspective of how we work together to validate our interpretation of the past. Not only because we come from, let's say, a UK school with some tradition that, they create a lot of the narratives, for example, in what is Egypt and all that area, Levante area. But with new techniques, can you continue with the narrative or can be displaced? So I think that challenge that in kind of situations.
So, do you have an example for that? I mean, have you found something that has challenged something that was established for in some place, and you found information that mm, it says other thing that
Let's
the
go, yeah, well, let's go with the same example of tobacco that we are talking about. So tobacco is a discrete topic that has been discussed in the archaeological and historical fields because it has social repercussions about, for example, hierarchy in society. Oh, only the poor workers were consuming tobacco in that way, or only the males were using tobacco, but not the females. And with these findings, we change that narrative. We found some truly molecular identification that say, okay, you need to think twice about that. If all the people are working, so how would the female were using tobacco? So how was that moment? Were hiding? Were because they will have second-hand smoke? Or those societies concepts about how we're working? So yes, that changed the perspective of how you look the past.
so I know this is a new field, so I don't know how many years the term has been established. So
4, I think, or 3.
so where do you see this field having the biggest impact in like 5 or 10 years?
This is starting to grow, and that's nice because more people is looking at the options to check different kind of materials. So as well, with this kind of webinars and seminars that we are doing, uh, the old leaders of the metabolomics and laboratories are opening the infrastructure to research. Because that was the big constraint in the past. Okay, you have an idea, you have whatever, but if you don't have the instruments, pues, nothing is going to happen. But now the laboratories are looking this as a feasible opportunity. So with good results. So in the next 5 years, I think that is going to expand the horizons in different materials. I know some of them that, but I cannot talk about that. I'm sure that it's going to be a nice way.
So my next question was gonna be so about see if you have studied another disease like Yersinia pestis, but if that is gonna be a sneak peek or something that you cannot talk about, I'm gonna keep my mouth shut.
In this moment, I am in a academic process in Colombia that after my work I returned, so I'm kind of a step away from laboratory. You know? So I'm not working directly with anything in this moment, but I'm participating as reviewer in different things. So there's a confidentiality in there, right? But I know that there is some nice things.
Thank you very much. That this webinar was very interesting. I think this is very new, but I mean you can get a lot of questions that maybe some don't have answers at the moment, but it's gonna drive you crazy sometimes, I think.
Yes,
Yeah, the
thank
most
you.
important is that you people from the life sciences see this as an opportunity and know about that. And for the reason I'm very grateful with Carolina, Juana, the Metabolomic Society, and Dr. Kala, because allow me to show this that, as you say, Juan, it's very new. It's very, very new. But knowing that exists is the first step to opening and to apply for that.
Yeah, and it's really good because we are used to think about the present and estimate the future, but not about the past. So it's a good way to see the the world, the people, the humans, and explore this field. So once again, thank you, Dr. Diego, for your talk today. And before we finish this webinar, I want to share the next webinar. So on April, we will have two professors from New Zealand. So if you want to join us on April's webinar, please register yourself on this link. You can find these links and all information on our social media. So please follow us on X and LinkedIn to attend this webinar. So thank you, everybody, and see you on April.
Have a great day, everyone.
Thank
See
you so
you.
much, everything.